genetic engineering. So microbes and disease. So due to microbes, what are the diseases caused by microbes? These are some diseases like pox, Ebola, cold, malaria. As we know, these few diseases has been known by us. So herpes and E. coli. E. coli also we know, HIV also we come to know about that. So thoughts for the day. So how can microbes benefit us in food and environment, in preventing disease, in agriculture, in energy, as well as in water waste treatment? So naming and classifying the microorganism. Linnaeus system for the scientific nomenclature. Each organism has two names, as we know: genus and specific epithet. And scientific names italicized and underlined. The genus is capitalized and specific epithet is lowercase. Could be as honor for the scientist. And the Latin origin, the Escherichia coli, that is E. coli, which is present in the intestine. Theodore Escherichia describe the habitat, which is present in the colon or an intestine. So Staphylococcus aureus, that it is a cluster and spherical or gold colored colonies, which is found in skin. So classification of bacteria. Bacteria, archaea and eukarya. So bacteria, archaea, which includes fungi, protozoan, algae, virus, and multicellular organisms. And eukarya, it's known as plants, animals, fungi, and protists. So bacteria. So now we are going to see about the bacteria, which category it belongs to. So bacteria belongs to prokaryotic organism, has peptidoglycan cell walls. These peptidoglycan cell walls, which involves positive gram-positive staining or gram-negative staining. We can able to identify the peptidoglycan cell walls by the technique called gram staining technique. This binary fission. Some few organisms follow binary fission, and some bacteria we don't. It can be a unicellular or multicellular that involves binary fissions, utilize organic and inorganic chemicals, and photosynthesis to obtain energy. So, for example, for photosynthesis to obtain energy, photosynthesis process takes place in algae. So, archaea, which is a prokaryotic organism which has lack of peptidoglycan. So, what is the difference? Bacteria and archaea. It has a peptidoglycan cell wall. It is a lack of peptidoglycan cell wall. So, live in extreme environment includes methanogens and extreme halophils and extreme thermophils. Thermophils, which is in thermal condition, that is heat condition. Extreme heat conditions. And type of eukaryotes. Eukaryotes, which is differentiate between protozoan and algae. As we saw, photosynthesis, which gains by the algae. Protozoans are unicellular eukaryotic and absorbs organic chemicals which are found in our environment. May move using pseudopods, cilia, and flagella. For example, pseudopods, we can say amoeba, which obtain the food from the environment. Actually, the movement of the amoeba is pseudopods. And the algae. Algae are the unicellular and multicellular eukaryotic organism, which has cellulose in their cell wall and gains energy through the photosynthesis. If we use salt water and keep it for 1 to 3 days, the water will start to grow in the water, the algae will started to go. You can, with the help of sunlight, with the help of photosynthesis process, it makes your own food, and the algae will be growing in the water. So produce molecular and organic components. So nowadays, algae becoming more popular in medicinal ways also and in food. It plays major role in foods also. And fungi. Fungi is the eukaryotic organism. So it has a chitin cell wall and use organic chemicals for energy. As we know, spores, we can able to find this chitin cell wall and the molds in the mushroom. Like you can say, able to see it in the trees. Some few mushrooms are growing, so forming a sporulum around the thing, and it will be getting burst. So molds and mushrooms are the multicellular organism, consists of mycelium and composed of filaments called hyphae. And yeast or unicellular. In fungi is eukaryotic other than yeast or the unicellulars. So virus or tools to be observed with a light microscope consists of DNA, RNA, or as a core. Is the core is which is surrounded by a protein coat called capsid. The core may be enclosed with the lipid envelope, and the lipid envelope is made up of proteins. And viruses are replicative only when they are living in a host cell. And the bacteriophage, viruses that infect bacteria, viroids, nucleic acid without protein coatings, and pyrons, infectious proteinous particles. Multicellular animal parasites, like we know about helminths, like flatworms and roundworms. These are called multicellular organism. You can see the flatworms and the roundworms. How do we view microorganism? When talking about the cell and the microscopic organism, you would be measuring like micrometer or stated in the micrometer. So as we know, to give a small idea about the micrometer, a human hair is 100 millimeter wide. A red blood cell meter micrometer wide. Typical size of animal cell would be 10 to 100 micrometers. So this type of microorganism, we can able to study about the different type of microorganism using this microscope. So there are different type of microscope, like light microscope, compound light microscope, and dark field microscope, phase contrast microscope, and differential interference, and fluorescent microscope, and confocal microscope. So these are the parts of the microscope, as we know. And this is the body tube of the microscope, and this is the auricular lens, that is eyepiece. It's the most important, which is able to see the microscope and adjusting the movement of the microbes. And this is the revolving nose piece, and this is the objectives. Objective, which was 10x, 20x, and 30x. We can able to see at the various concentrations of the microorganism at the various levels. And these two are called a stage clip, which able to adjust the slide, and diaphragm, and this is the light source, which able to directly falls on the specimen, and which able to clearly helps in viewing the microorganisms in the stage. And this is the coarse adjacent knob, and this is the fine adjacent knob, and this is the base, and this is the arm. And microscope is very, very important in microbiology. for the identification of microorganism and their function and their cellular movement, and finding whether it is a gram positive or gram negative uh, organism or not. So, compound like microscope. The image is magnified against by the auricular lens. So, the auricular lens is otherwise known as eyepiece. So, total magnification is equal to. So, this is a to find the total magnification. This is the formula: objective lens into auricular lens. So, resolution. Ability of the lens to distinguish 2 points, that is at the 0 point, and a nanometer can distinguish between 2 points to greater than equal to 0.4 nm. Shorter light wavelength produce greater resolution. So reflective index. In reflective index, light bending ability of the medium, and the light may bend in the air. That misses the small high magnification lens. Immersed oil. Immersed oil will used to help the air from the bending. So which we can able to see the microorganism clearly. And the type of microbes. So there are different type of microbes. And we saw the name of the microbe. Let us see the brief about the microscopes. So light microscope found in most schools for the identification of small organism or small insects or stain of the microorganism, etc. And use compound lenses and light to magnify object. So the lens can bend or reflect the light, which make the object beneath them appear closer. With the help of compound lens, we can use to magnify the object. We can able to adjust it in front. It will be able to appear closer, so we can able to see the microorganism so clearly. So stereoscope. These microbes allow for the binocular. There are two eyes. As we know, the binocular, we can able to see two eyes being of larger specimen. The spinning of microorganism at the top of this is a stereoscopic. And as we know, scanning electron microscope, SEM. It's the most important. It is used in mostly in biotechnology field to allow to see the movement of microorganisms clearly in scanning electron. Allow the scientists to view universe too small. It would be so small to see it in the light microscope, but in scanning electron microscope, we can able to see the movement as well as it is in as it is in large size. So SEM do not use light waves; they use electrons. So electrons is negatively charged electronic particles, as we know it, as we studied in chemistry as well as in physics too. To magnify object up to 2 million times. So we can able to magnify up to 2 million times with the help of these electrons. So transmission electron microscope also uses electrons, but instead of scanning, the electron passes through very thin specimens. So the specimen may be stained with the help of heavy metal salts. So magnification. So for example, your microscope has 3 magnifications like scanning, low, and high. Each objective will have the max magnification. In addition to that, the auricular lens has a magnification. See the magnification here, the scanning. So as we know, total magnification, magnification of the eyepiece, that is auricular lens, into magnification of objective lens, that is We can able to see the difference between that. Scanning is 4x and auricular lens is 10x. There is total magnification like we are obtaining is 48. So we can able to identify the magnification by with the help of this little small formula. So a brief history about microbiology and the development of microscope. Aristotle and the other believed that a living organism could develop from the non-living materials. Hans and Jansen's dash lens brand mounted two lenses in a tube to produce the first micro-compound microscope. And Robert Hooke, who published Micrographia drawing of microbiological material made with the best compound microscope and illumination system of the time. Anton von Leeuwenhoek was the first person to observe microorganisms, and the Carolus lens and Ernest Abbey pioneered development in the microorganisms such as lens and optochromatic lens, which reduce the chromatic aberration exist under the present day. Ernest Rose constructed a first electron microscope. So Anton von Leeuwenhoek described the bacteria. For example, he observed from like A and B are a rod form, and C and D are the motion pathway. A and B are the rod form, and C and D motion pathway. And E is a spherical form, and F is a longer type of spherical form. And H is in cluster. He proposed a scientific review to the Royal Society letter on September 17, 1683. He founded a simple microscope with one lens. See this diagram, which include the lens, black plate, and specimen mount here, and the screw plate, and focusing screw, and elevatory screw. So spontaneous generation controversies. So Francis, who was an Italian physician who refuted the idea of spontaneous generation by showing the rotting meat carefully kept from flies will not spontaneously produce mango juice. This is one of the major experiments performed by Francisco, which able to identify whether the meats can be whether in the meats the microorganisms are grown. As we can find maggots in some forensic investigations too, because after the death, during the investigation, many maggots will be there in the human. So, help of maggots, the full test has been done with help of it to identify some few clues also. So theorists helped reject the cell theory of the living organism, namely that all living organisms are composed of one or more cells. That cells are very basic functional unit of living organism. So Louis Pasteur is a famous experiments with Swanfish flag finally proved that the microorganisms do not arise by the spontaneous generations. So these are some golden age of microbiology because Robert Koch and Joseph Lister play the important role in identifying the microbes. So beginning of the Pasteur work include the relationship between the micro and the disease and the immunity and the antimicrobial drugs. So Robert Koch, who is one of the microbiologists who identified the bacteria which causes anthrax, and he experimented and introduced agar and transferred the bacteria with the help of inoculating loop to the agar, and he prepared a pure culture medium. So he introduced some coach postulates and the concept that a disease is caused by a single organism. And in Joseph Lister in 1865, he he also one of the microbiologists who has introduced the antiseptic technique and the use of phenol carbonic acid as a disinfectant. And Martin Bentrick, who discovered the viruses and the toxic poisonous infectious agents in tobacco plant fluids. And Paul Edsters introduced concept of chemotherapy. As we know, chemotherapy plays one of the vital role in the treatment of cancer. So use of salvage for the treatment of syphilis. And Alexander Fleming, who introduced who who first discovered the first antibiotic called penicillin. As we know, Alexander Fleming, who became the most important microbiologist in the field of microbiology. So Louis Pasteur in 1822 to 1873, he experimented with swan stick flax. He eventually developed for the sterilization and the development of antiseptic technique. He found a broth sterilized, which is trapped with air at the open end of the flask. And able to settle down the blend bacteria and dust. Broth allowed to cool slowly and broth stays sterile indefinitely.
निकिता साइलेंट कर लो भाई.
So flask is stilled so that sterile broth interacts with the bacteria and dust from the air. And when it's kept for an hour or a day, bacteria grows and multiples in the broth. So these are some proofs that microbes causes diseases. It was majorly from various microbiologists. As we know, Louis Pasteur, who proposed a germ theory of the disease, who believed that the disease results in divine punishment. And Pasteur fought for the surgeon that germ exists and carried disease, and dirty instrument handled spread germs. And pasteurization process kills germ and prevent the spread of the disease. And Joseph Lister, who introduced, as we know, antiseptic during surgeries. It is very important that we have to use antiseptic during surgery, because without using antiseptic, the germs in our hand will always affect the people who we are doing the surgery. By spraying carbonic acid on the surgical instrument so that it can be at the sterile condition for wound and dressing, he reduced surgical mortality during bacterial infection considerably. Robert Koch, who is a German bacteriologist, was the first to cultivate anthrax bacteria outside the body using blood serum at the body temperature. So these are some Koch postulates in 1884. The critical test for the involvement of microorganism in a disease. The microbic agent which may be present in every case of the disease, and it can be isolated and it can be cultured in vitro condition. The disease must be reproduced from the pure culture of the microbial agent, which is inoculated into the sustainable host. The agent must be recoverable from the external infected host. This eventually led to the development of pure culture technique and stains and agar and culture medium and petri dish too. This is one of the example that I am showing it in the image form. So for staining, for preparing a small for staining, and what are the essential thing like we want to know is for staining and coloring the microbe, they use dye to emphasize certain structures. A thin film of a microbe solution on the slide. A smart is used to fix to attach the microbes to the slide and kill the microbes. Staining. Staining, as we know about uh, gram positive staining and gram negative staining, and these have we can use with the help of basic dye and acetic dye. So instead, it consists of positive ions and negative ions. And basic type chromophore is in cation and acidic type chromophore is anion. Staining the background instead of cell is called negative staining. As there are different type of staining involves in microbiology. That is simple stain, gram stain, differential stain, and acid fast stain. So simple stain, staining with one dye. As we saw, it is like positive ion with the help of positive ion and negative ion. That is acidic dye with the help of negative dye. We can able to find whether it is an acidic or it is a positive anion or the negative anion. With the help of that, we can able to find that a micro whether the microorganisms are positive or negative. And the gram staining. Gram staining, as we know, I told about a gram positive staining and gram negative staining. We can able to find with the help of peptidoglycan cell wall. So, which the bacteria prone to penicillin and detergents. Positive gram positive bacteria are prone to penicillins and detergent, for example. For negative, it's more resistant to antibiotics. So, differential stain, it is a distinguish between the gram stain and acid fast staining. Differential staining. So acid fast staining, stain at the waxy cell wall is not decolorized by the acid color, and it is found in it can be used in microbacterium or nocardia. These are some special stains distinguish special part of the stain, like capsule, endospores. Endosol can be managed at green and saffron, and fragile are like carbophylls and simple stains. Like we can able to see the green format as well as saffron in color. So how to find distinguish between the positive and the negative? So as we know, gram positive bacteria prone to penicillin and detergent. So gram negative more resistant to antibiotics. So we can able to find the negative gram negative is in pink color and gram positive in blue color. So microorganisms something like study with the characteristic of technique include aseptic techniques and pure culture technique and micro. Is there any if you have any question, you can ask me in the chat box. So thank you guys for your patient listening. So if you have Any doubts, you can drop me a mail. That is drop me a mail to this mail ID. So that I can able to clear all your doubts. So what is the application
Friends, please fill up
of
the feedback form as well. I have given the link in chat. You can hold just a minute. I'm just sending you the link again. I think it didn't reach you all. I have set the link in chat. So you just copy paste that link in any browser and fill up the feedback form. If you face any doubt, please let us know. But please fill up the feedback form. That is very important for us to share the certificates as well.
Hello, ma'am. Meenakshi, ma'am. Hello.
Yeah, hi. Tell me.
Meenakshi, ma'am, I have a question.
Meenakshi, are you there?
Yeah, yes, ma'am.
Please continue, Meenakshi.
Yeah, ma'am. Question is like, what is the applications of microbes in food? Can you please explain in detail?
What is the
Hello,
hello?
ma'am. Can you get my question? What is the application of a microbe in genetics and food?
In genetics as well as in food.
Yes, ma'am. Please
There
emphasize
are
on
various simple, yeah. In food, for example, in food industry,
that.
like they are using milk to yogurt and yeast sugar to wine and the bread. They use it in dairy products like fermented milk, which is produced like inoculating the pasteurizing milk with a specific culture of the microorganisms. In your like we can able to do it in like for example in making of yogurt they're using specialized bacteria which can able to ferment and give some yogurts. So there are various processes which involved uh, in food industry. And moreover in genetics like we can able to distinguish the bacteria according to the various concern.
What concerns, ma'am? Hello.
The various application which is involved in like more like microbial genetics is mostly like a transmission of hereditary characters in microorganisms which plays a unique role in developing of ancient fields of micro molecular or cell biology. And major application we can find it in agriculture as well as in pharmaceutical industries also, which is mostly which is mostly interlinked with food as well as in genetics. And the most important advantage of microbes in the field of studying of genetics is uh, possibility of correlating the expression of gene which is intact in cells as well as in the expression in a system which can able to purify high purified compound in that microorganisms,
Hello,
and
hello.
will provide a simple system of health. Yeah.
Yeah, this is Virendra from AKS University. I just wanted to
Okay.
add something at your answer.
Yeah. Okay.
Yeah, ma'am. If you talk about the application of microbes in the genetics, you can easily go up with the RDT technology. They have wide applications of microbes, and apart from it, in any of the genetic studies, you can use the mortal organism as the microbes, because without microbe you cannot go for these studies. So it is the
Yeah.
backbone of genetic studies.
Yeah.
So I think I would answer this question.
Okay.
Okay. That is what it's backbone of genetics. You cannot remove out microbes for any of the R&D technologies with RDT.
Okay. And thank you so much for your reply.
Yeah, sure, ma'am. And even with the foods, ma'am, in case of food, there are wide applications of microbiology, because even if you are testing the different foods, like nowadays you are having the GMP foods, GMF foods, and you have spirulina as a food product for diabetic and all the nutrient concerns.
Yeah.
So
They're
nowadays
working with the
yes, yes,
they are
yes,
work nowadays they're majorly working with the spirulina and as well as
yeah,
algae.
exactly.
They are
Yes,
interlinking with the immunology also nowadays.
nowadays, yes, ma'am. So they are basically concerning, and with the applications of microbe, they are going ahead.
Yeah.
Okay, okay.